everyone. Hello everyone. Welcome to the live session for the Introduction in Cell Biology course. So we are here today to resolve some of your doubts and maybe help you recapitulate some of the points that we have discussed in the course. So as you all know, I'm Professor Shikha Laloria. We've been interacting via the lectures. So I will actually request my teaching assistants to introduce themselves also. They've been working quite hard in sort of helping in running of this course. So Debalina, could you please adjust your video, please? We can not see the whole face. So Ms. Debalina Dasgupta.
Am I visible or audible?
You need to adjust your camera so we can see your whole face.
Yeah, it's on.
The angle, you know, you need to, your face is not there in the field of view.
Okay. The webcam is on.
Yes, you can see your.
Okay, so maybe it's some internet issue. So,
Debalina, please adjust the angle of your laptop so we can see your full face. Yeah.
Is it fine?
Little bit more. Students would like to see us now, finally. Yes, very good. Wonderful.
Yeah. So,
Debalina.
hi everyone. So, this is Debalina Dasgupta. I am a teaching assistant for this course. I'm a PhD student in ma'am's lab. So, I will ask the other TA, Swagat, to introduce himself.
Hello everyone, this is Swagat Nath from Professor Shikha Laloria Lab, Biochemistry Department. So I will be the other TA who is assisting in this course. So if you have any questions on YouTube, you can post it in the chat box and we will try to answer it.
Right. So let's just explain the procedures. So there'll be a chat box in the Zoom as well as YouTube. And if you type your questions in the chat box, then we will be able to address some of them. At least we'll try to the best of our ability. And in case there are some questions unresolved, they could be addressed in the discussion forum also. So don't worry about that. So we will start trying to do that. So the point is that you have to type the questions. We already received some questions, I think, in the discussion forum and the other forum, and you can continue to post the questions also, and we will start trying to resolve some of them, and then we may like to revise some of the important points. Okay, so maybe let's look at, maybe there was a question about SMC proteins. Debalina?
Yes, ma'am.
I'd like to resolve that because some of the students were not able to understand the parts about SMC protein. So you can start and then I can continue also.
Okay, so I will first share the screen.
Yes.
Tell me whether you can see it or not. So yeah, is it visible? Hello?
Yes, Debalina, we can see that.
Okay, so
yes,
I will start. So let's start by understanding the structure of SMC proteins. So SMC protein stands for structural maintenance of chromosome complexes, and this protein has two coiled regions connected by a globular hinge region, and at the N terminal, also at the C terminal, there are characteristic Walker A ATPase domains which contain Walker A motif and Walker B motif. Now, each SMC proteins can fold back onto itself via antiparallel coil-coil interactions between these two coil-coil regions, and then it forms a rod-shaped molecule with the globular hinge domain at one side and the globular ATPase domain at the other side.
You lost audio, Debalina.
Proteins interact with each other at this hinge region, whereas in eukaryote there are six SMC different proteins which very specifically interact with their hinge regions, and then with association with other few components they make three complexes. These are cohesin, condensin and SMC56 complexes. So these all are representative diagrams for cohesin complex, condensin complex and SMC56 complex, and I will just briefly explain about this structure. So as you can see that in cohesin, SMC3 and SMC1 are the two SMC proteins.
Did audio lost?
Is it?
Yeah.
Am I not audible?
No, you are audible. I can hear you.
Okay. There was a technical glitch. I'm sorry about that. So let's continue. So in cohesin, there are two SMC proteins, SMC3 and SMC1, that forms the heterodimer. In condensin, there is SMC2 and SMC4, and in SMC56 complex, as the name suggests, the two SMC proteins are SMC6 and SMC5. Now, these two SMC proteins interact with each other at the hinge region, and these two are the ATPase domains. Now, there is another protein subunit which is called Cohesin subunit that asymmetrically interact with these two ATPase domain, and as you can see, it forms a tripartite ring-like structure. There are other regulatory subunits as well, mainly HIT subunits and then kite subunits that also interact with this Cohesin subunit and makes the holo complex. And as ma'am already mentioned in her lectures, the cohesin is mainly important for the sister chromatid cohesion, though it has a lot of other functions in maintaining the genomic integrity. Condensin, as the name suggests, it helps in chromosome condensation mainly, and SMC56 complex is majorly known to have its role in DNA repair. So I think that will be a brief introduction for you all for the SMC complexes. So I will now ask ma'am to move forward with the other questions.
Yeah. So I'll actually like to add to that because I think some students had certain issues, but we will be talking more about this family of proteins in the course. So basically in the lecture, I had introduced two of the complexes, the cohesin complex and the condensin complex. And if I remember correctly, there was a model showing how cohesin holds the two sister chromatids together. And so there are various models. One of the most popular models right now is that the cohesin forms a ring-like structure. And the two DNA molecules, they pass in between that. So it's referred to as the cohesin embrace model or the cohesin ring model, where it's actually encircling the two DNA molecules. So they cannot actually move away from each other. So, but when the separase enzyme at the metaphase to anaphase transition cleaves the one of the subunits, the MCD1 or SCC1 subunit, then the ring opens up and then the DNA molecules can come out and then they can separate from each other. So I hope that is clear to everyone. Now I'm seeing some questions here. So I want to clarify, many of you are worried about the exam and whether we have to read the links. Someone has a question about that. Let me just see. Yeah. So you have to follow the content being taught in course, and you might notice often in the last slide there will be a reference list. Very few, in fact, we are giving, but there are some which are listed as optional, so you need not worry about those. But the ones which are listed before that under references, it's nice to, help. It'll help you to actually go through them, because there'll be a lot of material available to not only which was in the lecture, but even more than that, if you all are curious to know more. Okay. So yeah, it's nice to see so many of you, so many of the students have joined today. So we are quite excited about interacting with all of you. So let me see what next. So somebody asked about PCNA. So PCNA, as you know, is a sliding clamp. Okay. So we talked about the E. coli sliding clamp quite a bit. And I did explain, but I'm happy to just mention this again. So somebody wants to know what's the role of PCNA in DNA replication. So it's a sliding clamp in eukaryotes, and it holds the polymerase onto the DNA, so that the polymerase, even if it falls off, like it's actually synthesizing DNA, but it could slip and lose the binding. And if it does that, then it will not diffuse away into the nucleoplasm or the solution around it, because it is attached to the sliding clamp. So the sliding clamp is sort of holding on to it, helping it, even if it loses the contact with the DNA and pauses the replication process or the synthesis of DNA, it's still in the general vicinity, and then it can restart. If PCNA or any other sliding clamp was not there, then when the DNA polymerase falls off, then it could diffuse away, and then the replication will continue when it comes and it rebinds, and that takes longer. So the sliding clamps, such as PCNA, they help in enhancing the processivity of the DNA. So DNA polymerase. So processivity means that once the DNA polymerase binds and it starts synthesizing DNA, how many nucleotides it can synthesize in one stretch without stopping. So that is what processivity means. So it enhances the processivity. Now, I think some of you want a question in the assignment resolved. It's something about, I think maybe the TAs can take this question. Actually, it's quite simple. So you can explain. One of you can go ahead.
Yeah, I will again
If
share
any
my
students want to know.
screen so that you guys can see this. Is it moving? Is the slide changed, Swagat?
I still see the SMC complexes.
Okay. Stop sharing. Share. It's just taking so much time. Yeah. Is it visible? Or am I audible?
We see the SMC slide.
Okay, so I think this is the question most of you have a doubt about. The question was, 1,000 base pair long double-stranded DNA molecule contains 30% adenine. So determine the number of guanine bases. So as you all know, according to the Chargaff's rule, the adenine and the thymine residues are equal, and the guanine and cytosine residues are equal. So I will just go through the basic calculation. So the percentage of adenine mentioned here is 30. So therefore the thymine is also 30%. So therefore the combined percentage of guanine and cytosine is 40%. So because guanine and cytosine residues are equal in number, so the percentage of guanine residue will be 20%. Now we mentioned in the question it's 1,000 base pair long double stranded DNA DNA, it's base pair. So if you calculate the number of bases, it will be 1000 into 2, that is 2000. So 20% of 2000 will be 400, not 200. So we, as an option, we gave 400 guanine residues. We didn't mention base pairs. So the answer will be 400 guanine residues. I hope this clears all of your doubts. And then over to ma'am.
Yeah, so let's check. Let's check if people have understood. If you all have understood, please say yes, understood, or still not understood. You can say that in the chat box, students. If you've understood the answer, yeah. All right, wonderful, thank you. Now, yes, so there was a question about that. There was actually confusion in one of the assignment questions, and we have actually announced that in the announcement section of the forum. But maybe we can just resolve this doubt, because there were two options that were correct. Okay, in the week 4 assignment, in one of the questions, there were two options which were correct. Maybe some copy paste error when we were making the choices. So what we have, the administration there, we have decided to remove that question. So your marks will be normalized from 9 rather than 10. So you won't be losing anything there. Okay, so that is the only solution that is allowed as per the software and other limitations which are there. So I hope everybody's clear on that part. Okay. And let's see what else we can address. Right, so I see one more question. So a lot of questions about replication. Indeed, it's very interesting. It was fun for me to teach it also. Like we all also increased our knowledge. So I'm happy you all are wondering about that and asking these nice questions. So one question is that how does the beta subunit of the polymerase increase the processivity? So a bit of a confusion. It's actually, there's a table in one of the, in the DNA replication 3. So beta is the name of the sliding clamp in E. coli. And as you all know, I mean, I just explained the role of the sliding clamp. So in E. coli also, so the sliding clamp is known as the beta, and it's going to hold the DNA polymerase in place, so it doesn't fall off. Okay, got that? Okay, so what else? I think maybe there are people on YouTube also, so we'll try to resolve as many as possible. And if there's something left, don't worry, I'll try to address it in the discussion forum also. Now, this question about CDC 25, I think we need to look into that. But I think I answered it in the discussion forum also. If not, so this is, of course, enzymes have specificities. And it's possible that CDC 25 recognizes, I mean, the region which is near the threonine 14 and tyrosine 15, and that's why it's able to remove that phosphate and not the other one, the threonine 161. But we certainly need to see if there has been new research in this field. And the other thing is, there was actually one paper that I had mentioned, and there's a slide in one of the presentations also, where we were discussing this issue, what is the effect of phosphorylation, and people had determined the structure. And on that basis, it seemed that the threonine 161, in the structure of the enzyme, it forms lots of interactions with the surrounding residues. So it's engaged and it's sort of buried, whereas these other two residues, which CDC phosphatase comes and dephosphorylates, they are more exposed in the structure, in the three-dimensional structure. So they are more accessible, and therefore they can serve as a very ready switch to alter the property. So that could be another reason that it can access these residues better than the other residue, which is already sort of involved in other interactions with other side chains in the molecule. But we can look up a bit more about it to be sure. But definitely this point was mentioned in that paper, and it's also there on the slide. So you can try to recap that. The questions will be, so you all are worried about the final exam, and I think the questions will be of similar type which are coming in the assignments. They will be objective type, and they will be MCQ type questions. So if you are doing well, or try to do as well as possible in the assignments, and try to follow the concepts also. It's not so important to memorize everything, but so the questions will be objective type. Okay. Yeah. I can maybe any of you wants to sort of participate, Deblina or Swagat, or should I continue?
I am not getting.
All right. So you can look it up. Meantime, I can continue. So I will like to bring up another question. So one question that had come up was in the forum, or I don't know where it was, but I thought it's interesting to bring it up because we are talking about DNA replication. And one of the students wanted to know, like, how does the primase know that, how many primers it should add, or where should it make the primer? Okay, I think that's the gist of the question. And you may remember that model, the loop model of the trombone loop model of DNA replication. I don't know if it's necessary for me to share the screen. Maybe I should do that. Okay. Debelina, do you happen to have that with you?
I don't have that with me right
Anyway,
now.
we talked about it at length, so I'm sure, I've brought it up many times. So the trombone model is that model where you see the formation of the loop on the lagging strand. And, on the leading strand, the primer, it is formed once, and then the enzyme keeps on extending it because there is no issue of directionality. But on the lagging strand, because it's in the opposite direction, again and again a primer is synthesized, and then an Okazaki fragment is formed. So the question is this totally random, or is the primase sort of preferring certain parts, or what determines the frequency at which the primase lays down the primers, and does it lay down at a specific sequence? That's the question. So I've looked up about this, and in fact, that's an interesting issue. So the primase, as you know, you would have seen that animation which was given to us by Dr. Drew Berry. The primase, in that, you will remember, it comes from somewhere, and then it comes and binds the helicase. It's not always present at the replisome. So it's not tightly associated with the replication fork. It comes and it associates with the helicase and the single strand binding protein coated single stranded DNA at intervals. And then every time it comes, it will synthesize a new primer. So this is because the interaction between the primase and the helicase, it's weak. And but after, if it forms that interaction, then immediately its activity, the primase activity, is stimulated by this interaction with the helicase. So when it comes and binds there, its activity gets stimulated by 1,000-fold. So it immediately lays down the primer. Okay, and then after the primer is formed, then the primase is released from the helicase. All right, so then the DNA polymerase can extend it further and form the Okazaki fragment. So the interaction is weak between the primase and helicase. If it was strong, then it would remain bound there all the time, and it would be hard for the polymerase to come and intervene and start synthesizing the DNA. So that's actually an interesting property that helps its function, right? So if it was stronger and there was a tighter association, then the primer synthesis on the lagging strand would happen more frequently, which is a question. And when there's more frequent priming, then the Okazaki fragments would be shorter also. So this weaker interaction results in the longer Okazaki fragments. And about the question whether, like, how does it know where to bind? One, it's actually coming and binding to the helicase based on its affinity for it. And that's a biochemical issue. But it also, in E. coli, people have been studying replication for many years. So a lot is known about it. And I came across some interesting papers. In fact, we have mentioned that by Yoda and Okazaki in MGG journal. And they studied this issue. Okay. And in fact, there is some small sequence specificity. So there'll be in E. coli, uh, the DNA replication initiation, it occurs at a specific sequence. Okay. So the primase recognizes the 3' GTC 5' sequence on the template strand. And but when the primase binds the DNA helicase, it actually, according to the authors, it releases the specificity of the primase. So primase can still lay down a primer, but it becomes less specific. So it's the sequence at which it can lay down the primer, that choice of sequences is broadened, let's say. That's one way to explain that. Okay, so now the consensus recognition sequence for the E. coli primase, which is associated with the helicase, is 3' purine, pyrimidine, pyrimidine 5'. So wherever that sequence comes, which is quite frequent, it will lay down the primer. So these are some interesting points about the frequency of initiation of DNA replication on the lagging strand. So that was nice, and it also got us thinking about this issue. It's quite interesting. I think what else? Is there something we can? Some questions, actually, if you just view the videos again, you will get the answer to. So we do prefer using the chat box, okay? Which of the following proteins? Okay, I have another question coming here. So there are some questions from YouTube, I think, also coming up. Right. Some of you had questions about the complementation groups, if I'm not mistaken. So complementation group is a very common term which comes up in the field of genetics. And so we came across this in the course when we were talking about screens, particularly I think the CDC mutant screen, where lots of different mutants were found by performing that screen. And I think in explaining that, I had mentioned two terms: primary screen and secondary screen. So one of the questions was, what is that? And I guess one way to address that is, doing the screen, it's a pretty big, it's a big job. You have to screen thousands of mutants, okay? I mean, in a normal screen, when you want to get interesting outputs, you might be screening minimum 30,000 or maybe even 150,000 different colonies. And so you need to have some smart way to, not only get mutants which are defective in the process you are interested in, but also smart ways to reduce your work while enhancing the chance of finding these interesting mutants. Okay. So what one normally does is, you will have a primary screen. In case of the CDC mutant screen, it was the temperature sensitivity, because it was thought that anything that affects DNA replication is going to stop the replication. So it's actually somewhat essential. So we won't be able to find a mutant. So we went for conditional mutants. So those scientists, they did a conditional mutant screen and look for temperature sensitive mutants. So that is the first screen. That is a primary screen. And that's easy to do because you mutagenize the cells, you add a chemical which causes mutations, and then you take those cells and you plate them for single colonies. And as I said, you will look at thousands and thousands and thousands of colonies. And then if you're looking for something like temperature sensitivity, it's quite easy to search for it because there's a technique known as replica plating, where you actually take the plate which has got colonies and you imprint it onto a velvet surface. And with that velvet, you can actually make prints of it or replicas of it onto fresh plates. And then those plates you can incubate at normal temperature. And when looking for a temperature sensitive mutant, you will also incubate one of the prints, one of the copies at a higher temperature. And then they looked for those colonies which survived at normal temperature, which is around 23 degrees centigrade. And those colonies, those particular colonies did not grow at 37. So then they collected those mutants from the plates which were at the permissive temperature. Okay, so a collection of temperature sensitive mutants was first made. That is the primary screen. Then among them, now one has to screen for whatever you are interested in, whichever essential process you want to study. In their case, it was cell division cycle. So based on the life cycle of budding yeast, they knew that, if there's a mutant defective in it, then it would sort of arrest with a cell cycle phenotype. So they had to observe that microscopically. All right. So yeah, this is question by Parth Ranjan. Somehow I see his name on the screen also. Right. So that's the primary screen is a collection of TS mutants. Probably they must have got hundreds of them. And then we have to see them under the microscope one by one. You put on a slide, or nowadays we have better formats also where one can see it, one can automate the process. But those days one had to look at it manually. So the secondary screen in their case was looking at the morphology at the restrictive temperature. So they took those mutants, those TS mutants, incubated them at the restrictive temperature, and then they looked under the microscope which of them are showing those typical cell cycle arrest phenotypes, which would indicate they have a mutation in some gene which is important for this process for regulating the cell cycle. So I hope that part about the primary and secondary screen is clear. And also the complementation group. So once you get a bunch of mutants from the screen, what are you looking for? You are looking to identify genes which might be regulating the process of interest. In this case, it is the cell cycle regulation. So we will get lots of mutants, and some of the mutants, many of them, they will have similar phenotypes also. For example, there was another question. There's CDC1 mutant and CDC28. And there's a data panel where I have labeled both of them the same panel. That's because their arrest phenotype looks the same. Okay. So that's a fine example about complementation group. So you got lots of mutants, let's say one which gives a CDC28 like arrest, that is a single unbudded cell with one DNA mass. And they gave them numbers. So you get mutants, you give them numbers. And then you assign complementation groups. And how do you do that? So complementation group basically is different mutants which are probably in the same gene. Okay. And I think that's what I tried to explain. Those mutations are likely to be in the same gene. And so out of any screen, whichever number of complementation groups you get, that is probably the number of genes mutated that you've discovered in that screen. But for each gene, there might be different alleles, okay, which have come out in the screen. So how do we do that? It's actually, I work on budding yeast. And these experiments were also done on budding yeast. So it's quite interesting to kind of explain that. So, budding yeast, it's exist in A and alpha mating types. So if you've done a screen where your collection of mutants are in A or alpha, or once you have done it, you can even transfer the mutation by crossing to the opposite mating type. Okay, so now let's say you have a collection of mutants in A and alpha background, you will mate them together and you will get a diploid strain, okay, which has got now one copy of each chromosome from one of the strains, which were both mutant. Now, if those mutations were in separate genes, different genes, then they are going to complement and the mutant phenotype will disappear, because if they are in different genes, means that each gene locus, it will become heterozygous. Okay, so and if the mutant is a recessive one, then this approach works quite well. And so this is how we assign the complementation group. So whenever you have two mutants showing a similar phenotype, you've crossed them, and in the heterozygous condition, the phenotype is remedied, then they are likely to be in different genes because they are complementing. But if the TS phenotype is retained, then you can think about it. It became homozygous for two different mutations in the same gene. So that gene product is not functional. So it will still retain the TS phenotype. And so those two mutations will fall in the same complementation group. So that's what a complementation group means. And I've also shared a link where, if you want to read about it, I think we replied to all. So you can read up at your leisure about this concept, but it's quite useful in genetics. Okay. Now, let's see what else is there. Maybe I'll check. So someone asked about intergenic regions. Any of the TAs want to contribute to that, or we need to? Intergenic regions, of course, the regulatory sequences are there. Promoters, enhancers, and insulators are also an interesting class of regulatory sequences. And then there's a lot of other DNA, and there are also non-coding transcripts, that is, don't code for proteins, but nowadays there are these long transcripts which have been found. And yes, there's lots of intergenic DNA. Some of it may have a structural role also in sort of organizing the genome. So anybody, Devalina, were you going to say something on that? Sagat?
Hi. So we are talking about intergenic regions. So basically, these regions are like those contains promoters, repressor regions, and activator regions. So these genes will involved in stuck. You stuck.
It's not.
Okay. So these regions are basically involved in regulating the transcription of genes. So, like for instance, sometimes CT, for instance, one of the protein CTCF and the other is cohesin. These two proteins are involved in like bringing these activator regions or repressor regions to the promoter region so that the specific gene gets activated or repressed. So these intergenic regions are basically involved in this regulation of transcription. Thank you. Over to you, ma'am.
Okay. So someone wants to know about how to frame a research question in the field of cell biology. That's a very general, interesting question though. So in fact, we have discussed the examples of the Hartwell lab. So you may be interested in a particular process, right? So you need to have assays, first of all, to look at that process. How will you study that process?
Yeah.
Whatever is unresolved, those are the good things to go after. You don't want to be left doing incremental work, just sort of repeating what others have done. So it's important to identify the good questions. And of course, you must have an assay to study your process at hand. And yes, perturbing the process to find the factors which affect it. And also another approach that I favor a lot is not to just stick with any one approach. So any question that you have picked to study, you must use a multifaceted approach. That is, use many different techniques. You can use genetics, biochemistry, cell biology, molecular biology, and also there are a lot of other techniques nowadays which have come up. So whatever are the current techniques which can help you in addressing that question, you must try to employ that. And so then you get a broad overview and the overall picture from all sides, um, to really solve the question. And any particular protein, what are its interactions with other molecules, and you should use different assays to sort of sort out, because some questions can be addressed only by genetics, some can be addressed by imaging, by actually seeing. Seeing is believing. We think something is happening if we can actually see that. So nowadays there are single molecule approaches. We've talked about cryo-EM, how solving the structure had given interesting insights. Of course, genetics is very important. When you have mutants that perturb a process, you can actually get to the mechanism very well when you combine that with biochemistry and cell biology. So that is something, all of you, I hope some of you will become scientists one day, and you should bear that in mind. And we've discussed many examples therefore of scientists and how they try to solve this problem of cell cycle. And I also discussed about Professor Howard Temin's work, where he was studying something which was of interest to him, how certain viruses, they infect chickens and they could transform cells. So how do they replicate? And out of that curiosity came out a very big discovery of reverse transcriptase in the field. And taking the observations, if the experiment is strong and we believe the result, then there has to be a rational explanation for it, even if it goes against the dogma. It went against the dogma of molecular biology at that time, that you can actually get DNA from RNA. So I think you all can. I think somebody was asking about that also. They may have missed it, but indeed that was a profound discovery, and it's a very nice example of how we are dedicated to solving one question, and it can really result in very interesting findings. And those studies, today funding agencies were looking at that. They would say, is this important to fund something which is affecting chickens? Why not study a human disease? But in those days, it was not so much everything translational for us. And that work went on, and but the discovery was such that it transformed the field of molecular biology, and it also helped us in facing the HIV. When it came up, it was a retrovirus. If people had not known that there are retroviruses and there was no history behind it, then when HIV came and it was infecting the populations, it would have taken much longer to understand its life cycle and to come up with interventions for HIV. So one should pick a question which interests you, and of course try to get funding, and nice people who are interested in sort of contributing to that work, and just stick to your guns. But you need an assay to study whatever you're looking at, and attack the problem from all angles. And that's what I would like to say about it. I see still, is there a problem still in understanding the answer to that question? Because I see more questions coming about that same question that Debelina has resolved in the assignment. Is the length between Okazaki fragments the same every time? So it won't be exactly, just as I explained, it's a process, it's a balance of the primase coming and binding the helicase, but the frequency at which it does it will depend on its concentration, secondly its affinity for the helicase. We discuss that, how it's weak binding. So at some frequency it will happen, but it won't be the exact same to the base pair. But roughly we have said that it can range from 1,000 base pairs to 2,000 base pairs in E. coli. and in other organisms, it's different. It differs between different organisms. So yeah, definitely the students are still wondering about the question. But I think we did resolve it. So I think we have also posted in the discussion group about that question. So I think we can sort of about initiation of DNA replication. I have actually taught in great detail. You can actually see both the DNA replication 3 lecture, and then there is also this entry into S phase, that lecture. It's all about the regulation, the licensing of DNA replication initiation. So I don't think we should repeat all of that here, but the videos are there on YouTube. So please look up these two. The one who has a question about initiation of DNA replication. There is a lecture about the regulation of entry into S phase and licensing of origins. So please look that up. All right, so let's see what else. There's an interesting question. I don't know if I'm supposed to answer it in this course, but someone wants to know, can extraterrestrial alien life forms have DNA as their genetic material? Now, of course, you know, if it's happening here, it's possible, probable, but I don't think anybody has found that yet. So we have to just wait, maybe, to see what interesting findings are coming out from that field. But yeah, if this can happen on one planet, surely can happen elsewhere also. And 10th question has been resolved. I think the textbooks, many people have questions about that. So TS, did you note down the names of the textbooks?
Yes, ma'am.
Yes. Could you share the information either here or on the discussion forum?
Yeah,
So,
I will drop the details of the books in the discussion forum. I think
okay.
that
So
way
I think you have actually answered the names of authors a few times, but I would like it if you were to tell the full name of the book and the authors and refer to the editions also. So if people have it in their libraries, or they could search, and you know, it will help the students to give the full title also.
yeah, sure. I will do that. I will do that in the discussion forum so that everybody can see that.
Need some water, please. Oops.
Okay, you have to first select the PowerPoint. कौन सा PowerPoint है करके देखिए.
This one. Is it visible there?
नहीं, ये 20 seconds लेट आएगा. नीचे नीचे.
Okay, I'm back.
It's live, Didi. It's live.
Can I stop share again? Huh? I can't hear.
नहीं, 20 seconds लगेगा.
Yeah, so here are the textbooks for cell biology. Students, Debelina is sharing this, I think. So
Yes,
you can please note it, take a screenshot or whatever you want to do. You all know that, no? You can take screenshots and save something that you want to revisit without having to watch the video again. So, I mean, we do that. So it's maybe worthwhile also when you're viewing the video, you could do that and save those. So when you want to review, you could actually go through that.
I will stop sharing the screen now.
Okay, I hope everybody has noted that. And Devlina, please share this in the discussion forum also.
Sure, I will do that.
Okay. There's something about senescence coming already. So I think I will also ask the TAs, or I hope this is being recorded. So please save these questions. Swagat and Devalina, you can copy and paste them. So if there are any left unresolved, we can look into that.
Yeah, of course, we will do that.
Okay. So nobody needs to worry. I didn't understand this question about the budding yeast short form code. Anybody wants to clarify that? Can budding yeast short forms code? We have to remember. I'm not so sure about that. Anyway, so budding yeast, the scientific name is Saccharomyces cerevisiae. So you may have seen this. Sometimes I refer to it as budding yeast and sometimes as Saccharomyces cerevisiae. Maybe that's what the question is about. I think P53 will be discussing more in upcoming lectures. And we cannot share the textbooks, that's they are by publishers. So yeah, genetics books are fine, and you definitely can look into genetics books for all this complementation and screens and mutations, even something about DNA replication you may find there. So we can share more references. Some of the references that we share in class, let me tell you, in the last slide, they're the optional ones, and the ones which are listed as at the top, you can read it, and many of them, they are actually freely accessible on the internet. So if you type the title of that paper, it might start coming up. So I'm trying to be careful about that, like the compulsory references, I'm trying to make sure that they are free access or open access for everyone. Okay. So that's something, I don't know if how many of you went and looked up the review articles or papers that were suggested. Senescence genes. Okay, somebody is asking a question from Debolina, a student, not the TA, I suppose. So actually a lecture about senescence is coming up. So you can wait for that. I think it will be next week. We have prepared one lecture on senescence, so we can maybe look into that. Telomerase, we did talk about telomerase quite a bit. And basically, it's going to synthesize a primer. I'm actually not able to share my screen because I have to refer to many things here. Let me see. But this part was covered a little bit in the class, I think. So telomerase has got RNA also. And in fact, some of the groups, they actually contribute to the process of the synthesis of the primer. So it provides the primer. It's part of that enzyme itself, and then it synthesizes also. So if people are more interested in reading up advanced aspects of this, we can actually find some references and share with you. But for this course, I thought whatever I taught is quite sufficient. But of course, there's no end to knowledge, so all of you can look up more about the topics. Okay. So, but telomerase also, it's a ribozyme because part of the RNA contributes to this catalytic process. Swagat, could you please save the chat? So if I'm sure there are questions we have not answered, or if we have missed it, no, we can actually look into that. You can just scroll, copy, paste the chat. Click on the chat, it'll come on the side. Maybe,
yes, ma'am.
yeah, so I do that. So in case we have missed anything, and I'll check some of the other places from where the questions are coming, because the session might end actually. I myself am doing that. Okay. Yes. Albert's and Lodish both are good books. Somebody is asking which one should I go for. So they are both good books. I cannot pick between them, but yeah, they are both, they have complementing material, whichever ones you can get your hands on. I can say though that one old version of Albert's is available online. It's quite old, but it's got useful information. So you all, those of you who don't have the books, you could actually access that. You can't download the whole book, but you can search for specific topics and read them. So maybe we will share that also. Both these books are excellent books by Bruce Alberts and the book by Lodish. They are very good. Okay. Will we have another meetup before the exams, final exams? Yeah, we are hoping to have at least one more, if not two live sessions. So we will talk to our administration about that and all that. Some are questions, some kind of advice, I would say. Being in IC, I would say, yes, IC is great, and other institutes in India also very good. We're all proud of our scientists in IC, in TIFR, in various universities everywhere. So, you can look up the research work and choose accordingly, right? I encourage whoever is interested, please, we would like people to enter this field, and if not, at least enhance your knowledge about cell biology. And I think Shashwat wants to know that biological sciences, TIFR or IC. So try your best for all of them, both excellent institutions, and there are more excellent institutions also in India. We are doing very well. A lot of new institutions have come up, IITs, universities, many institutions are there. So, nowadays you all are blessed to have the internet. So you can search and, follow your interests, whatever interests you in research, and follow your dream. That's all I'll say. All the best. Thank you everyone for coming for the session. We had a good time, and I hope I was able to resolve the doubts. And if there's anything remaining, we'll try our best to come up to the expectations. And also, yeah. Okay, so we can continue to interact in the discussion forum. Shall we close the session now? I think time's up. Okay, thanks all of you for joining. It was wonderful to connect with all of you, and we will try to connect with you at regular intervals to resolve the doubts, and please do interact in the discussion forum. Thank you. Bhagat, could you end the meeting now?
Didi?
Yeah, she's coming.